If I reach into your brain right now and pull out your right hemisphere, you would die, right? Most people would die. If you do that in a developing infant, they would actually still survive. And he had this really beautiful experiment where you have this sea slug, and some people think, what can we learn from an invertebrate organism? And we actually learned the entire mechanism of how memory is formed. There's visual learners, there's auditory learners, and that there are kinesthetic learners. That is more or less completely incorrect. And I think is very misleading to both educators and to students.
At like the levels of neurons and circuits, what even is learning? Why can some students hear something once and remember it, while others need repetition to do the same exact thing? Why does learning an instrument change your brain differently at 16 versus 60? What's physically happening inside your brain every single time you learn something new? These aren't just study hacks or productivity tricks. They are real biological events. Neurons are rewiring, synapses are getting stronger, and finally entire circuits are reorganizing who you are. Hello, I'm Avnish, and this is The Early Perspective. Each episode, we break down how the brain works and what that means for how we learn, think, and live. This week, we're going to be talking about what learning actually is on a neurological level. We're also just going to be talking about how it is studying neuroscience from the inside, including the research, the culture, and just the reality of life as a grad student. To help us do all of this, I'm joined by Lucas Encarnacion-Rivera, a neuroscience PhD researcher at Stanford University. He also did his undergrad in neuroscience at Emory University and was trained to be a researcher at the Princeton Institute of Neuroscience and the University of Pennsylvania. Lucas, thank you so much for joining me here today.
Thank you for having me.
I just wanted to start with you and really your experiences. Could you first tell me about why you're just researching and studying neuroscience?
Yeah, so back when I was in high school, I thought very intently about what sort of study I wanted to pursue. And I was actually initially drawn to medicine, specifically neurosurgery, after I had read Ben Carson's memoir. And after that, I started thinking more intently about research and neuroscience. Really, how do we come to know the things that we know about life, about the brain and nature? And I saw neuroscience as this really cool window to answer some of the most existential questions to humanity. What is the nature of emotion? What is the nature of consciousness, right? What is the meaning of life? How does the brain manufacture happiness? And I felt that neuroscience was actually poised, especially in the 21st century, to give really convincing answers to these questions. And so, because of that, I knew I wanted to pursue research in neuroscience. And then, as you had mentioned in that kind of introduction, went to Emory and then worked additionally at some other places to really understand the brain. And so now that's all brought me to kind of being in my PhD here at Stanford now, sort of at the latter stages, trying to wrap things up.
I also absolutely love neuroscience and do believe that it could help answer many of these existential questions that you're talking about. So, if that really is your goal, what is the core of your research? What are you doing right now at Stanford?
Yeah, so I have two primary projects at Stanford. Both of them are slightly different. The first is trying to understand how the brain actually encodes and represents homeostatic drive. Really, when I came to Stanford, I was interested in what is a subjective state in the brain, right? Think of emotions or think of
Mm-hmm.
feelings. And probably the most primitive of these is hunger and thirst. In fact, I like to think that the first ever thought of a living system was probably something like, I am hungry or I am thirsty. And so, because of this, organisms have developed these fantastically complex mechanisms that are conserved across many of the mammalian species to really regulate the balance of energy and water. And a lot of it starts with this little pang in the stomach or kind of the dryness in the mouth that all of us uniformly experience day to day. Um, and really, in my opinion, lay at the bedrock of conscious experience. And so, what my work sort of initially entails is really trying to understand how these feelings, hunger and thirst, are actually encoded in specific neurons across different regions of the brain, and how these are actually mechanistically related to actions. So, if you feel hungry and eat, that causes you to go eat something. The second project I'm working on, actually sort of unrelated, is how a movement disorder called tardive dyskinesia progresses and exists also in the brain. Typically, when people present with psychosis, schizophrenia, or other neuropsychiatric conditions, they're given a class of drugs that acts on a dopamine receptor. Now, people very famously know dopamine for its involvement in reward, but dopamine is also incredibly important for movement initiation and suppression. In fact, Parkinson's disease, which many people know, is caused by degeneration of specific dopaminergic pathways in the brain. And persistent use of these antipsychotic drugs in perfectly normal patients can eventually lead to a motor disorder characterized by this aberrant movement of the face. And there's been a lot of hypotheses as to how this actually transpires and what are the neural mechanisms at play, but there hasn't been any real robust and in-depth characterization as to actually what's happening. And so part of my research is to investigate the actual neural dynamics in animal models that is causal for tardive dyskinesia, as well as trying to elucidate sort of potential new therapeutic targets, whether it's druggable targets or, for example, neuroanatomical targets for deep brain stimulation to sort of ameliorate symptoms for people living with this disorder.
Yeah, I think that's honestly wonderful that you're doing such complex and very diverse research. Much of my research was mostly on neurogenesis and how it could be used as a potential therapy for neurodegenerative disorders. So that alone took up a lot of my time and made my day very complicated. So, how are you able to balance both of these very diverse topics of research? How does your typical day look like as a PhD student?
Yeah, so I'm at the intersection of systems and computational neuroscience, which means that day to day things can vary. Really, I like to describe them in sort of epochs of work. So there'll be periods of time where a lot of what I'm doing is predominated by surgeries, running experiments, which it could be behavioral experiments, could be experiments where I'm recording from different regions of the brain, or as I mentioned, sort of doing these micro scale neurosurgeries in the mouse brain, whether it's to implant electrodes or to inject viruses that sort of deliver genetic payloads of interest. Aside from that, I also sort of develop and analyze data, and that includes a lot of coding, a lot of data analysis, a lot of thinking about how to model neural circuits. And so in those epochs of time, I'm spending a lot of my time in front of the computer, looking at matrices of data, trying to extract meaning from the experiments that I had run. Nowadays, I've sort of finished a lot of my experiments and analysis, and a lot of my days now are just focused on writing my papers to prepare them for publications. So day to day varies, but I'd say those are like primary genres of what day to day can look like.
I think that's yeah, I think that really puts things into perspective about how different like there's a lot of differences in how a typical day is, but also how your research breaks down and how you're managing all of that. I think that was very important. Thank you for sharing that first of all. So I think with that, we can get into really the crux of our conversation today. That is going to be the science behind learning. When most people think of learning, they are picturing flashcards, lectures, and repetition. Personally, whenever I'm thinking of learning, I'm thinking of a classroom with a teacher lecturing or just late night reading of a paper or a textbook. All of this is really just what comes to mind when I think of learning, but of course, it isn't an objective definition. So, how would you define learning at like the levels of neurons and circuits? What even is learning?
Yeah, I think this is a fabulous question that has really important implications across biology and psychology. I'd say in its simplest form, learning is the change of synaptic strength between groups or ensembles of neurons. Now, everyone knows that our brains are composed of neurons, and there are connections between these neurons that are called synapses. And what learning is essentially that the sort of strength between these connections ultimately over time changes. And that can look manifest in a lot of different ways. It could be that they become really strong. It could be that they become less strong. And when I say strong or less strong, what that actually means is if neuron A fires, what's the probability that neuron B fires to which it's connected in this very simple two neuron circuit? And what learning could look like in that very simple two neuron circuit is that there are changes between neuron A and B that mean that when neuron A fires, the probability that neuron B fires starts to increase. So that's sort of what it is at a fundamental level. Now, how this actually plays out in real systems, I think could be really illustrated well by Eric Kandel's famous work in aplesia, which earned him the Nobel Prize several decades ago. And that he sort of was able to find what is the neural basis for a memory. And we sometimes think of memory as distinct from learning, but in fact, memory is just the sort of actual implementation of something that is learned, right? So memory is a learned bit of information. That could be a name, that could be a place, that could be a skill on the guitar, for example. And he had this really beautiful experiment where you have this sea slug. And some people think, what can we learn from an invertebrate organism? And we actually learned the entire mechanism of how memory is formed. Uh, and you have this small organism, and it has this siphon that it uses for a variety of things. Now, because the siphon is important, if you poke the siphon, it'll retract, right? So this is a sort of protective mechanism for the sea slug. Now, if you keep poking the siphon, eventually it'll habituate, meaning that it realizes that this initial poking of the siphon is sort of not that important. Now, if you then reintroduce a shock and poke the siphon, so that meaning when you poke the siphon, you're going to induce an initial shock, then the siphon constricts even more. So ultimately, what this is to say is you have this system where this sea slug can remember what has happened to it in the past. And they were able to, what sort of Eric Kandel and his lab was able to accomplish was they're able to map the circuitry of this memory of sort of I've poked your siphon in the past and now I've shocked you. And they were able to find that the siphon has sensory neurons that map directly onto the motor neurons that are controlling the siphon. And through that connection and the strength, the relative strength between those connections, they were able to modulate, or the sea slug is able to modulate the extent to which that siphon constricts. So that's sort of an example of what I'd say is, in sum, is that learning really is just a change in strength between circuits, where strength is really a measure of probability between firing. And sort of a very simple implementation of this can be found in animals where you have modulation of specific behaviors, whatever it might be, by certain inputs.
Right. I think that really helps explain things a lot and puts things into perspective, especially with the experiment that you talked about with the sea slugs. I
Mhm.
really didn't know that they use the sea slug to help map many of our, like, a major part of our understanding
Yeah.
of memory and how the neurons are working. So I think with that groundwork laid, let's dive even further. So learning generally involves neuroplasticity is kind of what you already touched on.
Hmm.
So from my understanding, that is mainly defined as your brain changing and new connections forming between neurons every time you learn or try something new.
Hmm.
I'm often at elementary schools trying to give presentations introducing them to neuroscience. So whenever I go over learning, the question of the science behind it which is of course neuroplasticity, is one that keeps coming up. Obviously, I don't want to scare them, so I'll explain it in really simple terms. So, could you go into a little more depth on that? What exactly is happening with
Mhm.
neuroplasticity? What does it even mean?
Yeah. Yeah, it's cool because I think plasticity and synaptic plasticity, as you point out, have become these buzzwords, and
Mm-hmm.
people sort of throw it around without actually understanding what's taken place. Now, the field of synaptic plasticity is incredibly large, but a lot of the work is owed to Rob Malenka, who's actually still a professor here at Stanford. And he described a lot of fundamental mechanisms for what I just alluded to earlier, right? You have, neuron A and neuron B, they're connected, and there's a change in the probability that if neuron A fires, that neuron B is going to fire. And a lot of this is based off of, well, sort of Hebbian learning rules. And a lot of people have probably heard of this, but this is that neurons that fire together wire together. And this is sort of classically attributed to the famous neuroscience, Teb. However, it was actually said by Carla Schatz, who's a fantastically talented neuroscientist and sort of titan in the field, also here at Stanford. So neurons that fire together wire together. What does that really mean? It means that if neuron A continues to fire to neuron B, that eventually these are going to undergo plasticity such that now if neuron A fires, the probability that neuron B fires increases. Okay. But now you ask, I think, a very insightful question, which is what is actually happening between neuron A and neuron B that's causing that change in probability? And what's happening at a very fundamental level is that sort of neurons communicate through voltage. And between synapses, there are actually sort of a transfer of chemical information that binds onto neuron B that then changes again the sort of electrical potential in neuron B. And what's interesting is that there are certain classes of receptors, sort of NMDA receptors and AMPA receptors. Now, the names of these don't matter in as much as they are just sort of nomenclature for characterizing them. And NMDA receptors are usually blocked by a magnesium ion. Now, magnesium, it's a very large ion, and so it blocks the pore of this channel on the plasma membrane. But what happens when neuron A is firing a ton and it's delivering a lot of neurotransmitter, and that's causing a lot of increase in the synaptic potential in the postsynaptic neuron? That results in the very positively charged magnesium ion to leave the pore. And so NMDA receptors open, and then that allows for the influx of calcium. Now, calcium is actually kept in extraordinarily low concentrations inside of neurons. And the reason that's important is because calcium acts as a second messenger. Usually, calcium will bind to specific proteins, and that leads to sort of activation of calmodulin or other calcium binding proteins. But what's really important here is that the influx of calcium results in an increase in transcriptional expression of another receptor called AMPA. And AMPA is this excitatory receptor that binds glutamate, which is one of the primary excitatory neurotransmitters of the brain. And so now you have a situation where AMPA is being sent to the membrane, and that neuron now becomes hypersensitized to input from neuron A. So that, in a very sort of basic way, is sort of how neuroplasticity works.
So, I think another buzzword which is very commonly mentioned is the brain's wiring and how it's rewiring.
Yeah.
How literal is that? Is there any sort of change in shape and structure when we talk about that?
Yeah. So the brain does literally rewire. I just mentioned how, for example, there are changes in the composition of receptors that are
Mm-hmm.
expressed on the plasma membrane of cells that make them sort of more or less receptive. But there are also physical differences. In science, we call these morphological changes. And that could look like the actual shape or size of the dendritic spine. There's a whole subfield of neuroscience dedicated to this. And it's really beautiful. You can actually express a fluorescent indicator in an axon and its dendrites, and you can see in real time how these synapses are extending, receding, and those are sort of the literal physical qualities of how neurons wire themselves. Additionally, there's actually very, very famous work from one of my co-advisors, Li Chen Lo, here at Stanford. And his work, part of his lab, is dedicated to understanding how genes can wire neural circuits. And they do these beautiful experiments in flies where they're able to alter the expression of certain genes, and they can literally use those to rewire different neurons to each other. And when I say rewire, I literally mean neuron A used to be connected to neuron B, but I'm going to change a gene, and now neuron A is connected to neuron C. And they use that to sort of understand the sort of genetic and functional logic of how neurons wire during development. Now, these might be all very abstract examples, but a very sort of intuitive and interesting example I like to give when I receive this question is if I were to look at a region of the brain called the somatosensory cortex in me, for example, I don't play violin or at really any physical instrument other than drums for that matter, versus a professional concert violinist, and you were to scan both of our brains, right? What's fascinating is that the brain of the professional concert violinist is going to have a greater volume in the somatosensory cortex that's, for example, dedicated to their left hand, or whichever hand they might. And that is a sort of literal and very visual interpretation of, okay, this person has practiced for thousands of hours with a certain hand, and that has then led to, in consequence, an increase in actual volume, which corresponds actually to the sort of density of synapses, for example, representation of that hand in the brain. Another really important example of this, or interesting example, is if you were to look at the volume of the hippocampus, which is a region of the brain that's involved in memory, in me, who am just a normal dude, versus taxi drivers in New York, who, of course, have to have an extraordinary spatial memory. What you find is that the taxi drivers have huge hippocampi, and the volume of sort of the volume of the gray matter in the hippocampus is actually positively correlated with the number of years that they have served as taxi drivers. So the most experienced, right, the best spatial maps in the brain happen to have the greatest. Um, so that might be more intuitive examples of what
Mhm.
this actually literally manifests.
Yeah, I think for sure because the examples you gave helped really make it less abstract and more of a an idea
Hmm.
that people can digest. So
Hmm.
at least to sum up a lot of what you're saying is that there is literal change when you are learning, and then synapses and the connections, all of that is changing. So
Hmm.
how exactly is that affecting our memory? So let me just go on a tangent for a little bit. So I remember my childhood home had this huge library filled with dusty old books. I read a few of them, but most of them were just tucked away. So whenever we cleaned up the library, I just pick up a random book and it had a dusty smell being let out.
Hmm.
That smell would instantly bring back all the memories of me reading the book and how it made me feel. It's kind of like how you when you get into a new car, you have that new car smell where that brings back the memories of when your car was new. So, how are smells such great triggers of memory? Why was I able to recall such old events with detail, yet information from textbooks I just read last night seemed to elude me?
Yeah. So this is a really fascinating question, and I think there are some answers, and there's a lot of work that's gone into this. What I'll say is olfaction, which is just a sort of term for smell, in addition to other sensory modalities, happen to be really good at sort of eliciting memories, mainly because of the salience of that. So, what do I mean? Well, the brain is really good at identifying contexts that are relevant for the future. So, let's say you're a predator in the wild, or a prey, more importantly, in the wild, and you know that if you smell a certain thing, let's say another predator has been here. That's a really good indication that there could be, and let's say you've had some experience with predators in the past, that's a really good indication that there might be a predator nearby and you should probably flee. And so the brain has become really adept over sort of millions of years, billions of years of evolution to really understand and internalize certain sensory contexts. Now, why olfaction? I think olfaction is especially interesting because as a sort of sensory modality, we're able to discriminate between hundreds of thousands of individual molecules that are present over everyday life and everywhere that we go. Now, so olfaction happens to be more important in other species, like mice, for example. Um, and a really good example of this is there are memories in mice that there are, how should I put this? Mice have memories of things that they have never experienced. So that statement might be kind of counterintuitive, because we all think that memory is derived from experience. But a perfect example of how this dovetails into olfaction is if you put a mouse in a room, and on one side of the room you put a droplet of cat urine, or let's say, or for example, you have a piece of bedding and you allow the cat to walk on this bedding. The mouse will, even if it has never seen a cat in its life, even if it has never smelled a cat in its life, it will always avoid that area. So that is a sort of an example of this sort of deep evolutionary urge to, for example, in this case, avoid a predator. And there's sort of work done by Lieberless out of Harvard that describes a specific subset of olfactory receptors that binds to specific amino acid derivatives that cause this. And so that's, for example, an example of how olfaction is so potent that it can trigger memories of things that you have never experienced before. Now, we don't have evidence of this, for example, in humans as of yet, but there are probably some examples of this. But that's a very clear one.
So you're saying that evolution has been like one of the primary contributors to why olfaction or just the sense of smell has been such a big trigger of memory, right? So are there any other sorts of explanations for why olfaction is so important?
Yeah, so part of it is you want to really think of olfaction is not only important, right? So in humans, we're a visually dominant species. In fact, most primates are. If you look at, for example, the photoreceptors that we express versus mice, mice are notoriously they're not good visual species. So a lot of it goes back to is this German word called Umwelt, which is what is the totality of a sensory experience of a specific species. And that looks different, right, for us versus a bloodhound, right? What is it like to be a bloodhound? We can never really know because they have olfaction that far surpasses ours. So part of it is that there's evolutionary pressure based off of specific sensory modalities that animals are attuned to. But humans, for example, we are really good at analyzing faces in a way that no other species, except, for example, some primates can. And if you see a sad face, even if it's for a fraction of a second, you can instantly ascertain the emotion of that person was experiencing. And so it's not only olfaction, it just so happens that, for example, in mice, they have had that as a dominant sensory modality. But in humans, that's also, there are others, like namely vision, that we are supremely adept at. And that's primarily a function of evolutionary pressure.
Right. I think that was honestly a very big question that many people had because
Hmm.
memory and learning in general just are quite confusing and has many parts of it because
Hmm.
many of its parts are just widely accepted and not really questioned. Another very well accepted fact is that memory is is greatly enhanced when some event is traumatic or just emotionally important, and people tend to remember those kind of events in very vivid detail. For example, my sister always talks about how she remembers in vivid detail too about when she was 3 and dislocated
Hmm.
her hand. She talks about how her friend pulled
Yeah.
her hand too hard and the exact place, the time, everything. That was almost 20 years ago, but that was when she was 3. I remember nothing from when I was 3.
Yeah.
So, how does that work? Why is the brain prioritizing such traumatic or important events like that?
Yeah. So the intuitive explanation is because they're relevant, right? If I have a traumatic event that has happened to me, right, my brain is going to tell me, okay, this is really important, right? If you can think of again in a state of nature, let's say a mouse, right? Knowing the difference between cat urine and another species urine can literally mean the difference between life and death, right? So the brain over, these billions of years of, or hundreds of millions of years of evolution has become extraordinarily good at pattern recognition. Now, what's more substantive question is what's actually happening. What really is sort of what I can sort of describe as being fundamental is there are circuits in the brain that recognize valence. Valence is this kind of continuum of pain or aversive stimuli to pleasure, right? So pause something with positive valence, right? For me, maybe like a cup of ice cream, right? Negative valence could be, all right, now someone has pulled your finger. And there are circuits in the brain, specifically the amygdala, for example, that can determine the affective or emotional salience of specific stimuli. And the amygdala can, in turn, communicate with other brain regions to essentially enhance the relevance of that memory, which then leads it to becoming it leads it to becoming more entrenched, easier to recall. An example of this is if you take a mouse, for example, and you put it in a room and using the olfactory context or visual context of the room, it can understand where it is. Then let's say you deliver a mild electrical shock, so it's very negatively aversive to the mouse in this room. Then you wait a day. The next day you put the mouse back in this room, right? What happens? The mouse freezes. It becomes really scared because it knows yesterday I got shocked and I was in this environment, and so therefore it's very likely to happen again. Now, if you, for example, if you then deliver a virus that has this specific protein that allows us to control the activity of neurons with light. This is a method developed actually in Carl Dijkstra's lab, the lab that I'm in right now, called optogenetics. If you now deliver light to, for example, the amygdala or other brain regions that are participating in this memory formation, you are able, in the absence of any context, to induce freezing. So let's say the mouse is not in the room, it's in its home cage, and you activate these neurons, right? That you are able to now restore that memory. Um, Another really good example of this is that if you, let's say something with positive valence, right, instead of foot shock, if you put a mouse in a maze and it has to find a reward, well, in nature, you want to know where specific resources are. And actually, I think I'll touch on this after if you talk about sort of dopamine and emotion and memory.
Yeah. So, do you think which is more heavily weighted? Is negative valence more important and prioritized by the brain for memory? For example, if there's a traumatic event versus some super happy event, is the brain more likely to remember in detail, especially the traumatic event?
Yeah, so part of this is contingent on stage of life and like the actual nature of the traumatic event. Um, I don't want to speak. I don't want to speak from authority here. I have not read a study that has demonstrated this. But at least from my intuition, I think traumatic events tend to be more salient. Um, but it's all along a continuum, and it's like how do you really quantify how traumatic something is versus how valuable something is, right? If you look at like the yin and yang, let's say, if a traumatic event is, at least in mouse, people simulate this by taking a mouse from its mother and having it reared in alone, right? This is very traumatic for animals. They're cold, they don't know what they're doing. Similar to babies, baby mouse pups are very helpless in the world. Now, what's the sort of positive valence counter of that? It's very difficult, I think, to quantify. So I hesitate to give a direct answer to that question.
Yeah, I think intuitively what you're saying makes sense, but I
Hmm.
get how it would be very hard to quantify something which is
Yeah,
very happy versus very dramatic,
yeah.
because especially it's a very subjective thing in general.
Hmm.
So let's let's talk about something less uh like not as traumatic, but rather if people choose to not care about something, like for example, if a student says, I don't care about this class, so I can't remember anything, is that a genuine neuroscience problem, or is that just related to motivation?
Yeah, I guess, are you saying is it more about the ability or about the willingness?
Yeah, exactly.
What I'd say is there's a spectrum of people's propensity for, to do well in certain subjects, right? Some people can look at certain mathematical, like I have some that are behind me. I didn't write these at all. Um, and there are some people that can look at this and very intuitively understand what's going on, and there are other people that struggle. And so there are naturally, yeah, of course, there's going to be a sort of spectrum of how tuned people are to particular subjects. Now, that being said, I think learning, this is more psychological as opposed to neuroscience. But psychologically, motivation has a very clear role in how well people are able to internalize information. So if you tell yourself, right, I don't want to do this, and you don't actually study, then it's possible that you're just it's also possible you're just not going to do well, and that actually has nothing to do with sort of your ability, which acts as really more of an initial condition. And so my sort of belief that I think is probably rooted in a lot of psychological evidence is that baseline ability that we can see as operating on some distribution really acts as just an initial condition, and for the most part it's really up to the motivation of individual people to, and also the studying style, right? Because there are, I think, certain ways of internalizing information that are better than others. Um, it's really up to that in person to actually be able to do that. So I think it lends itself more to motivation. That being said, I don't know necessarily the neuroscientific equivalent of this, so I hesitate to speak authoritatively on this subject. I think it's more of like a educational psychologist would probably be more poised to answer that question.
So I know that you touched on how different learning styles can also contribute to how people retain memory. So then,
Hmm.
is what is the science behind that, and what would you suggest to somebody who's trying to really learn something better?
Yeah. So I'll give an answer to this that I think is, it's not actually going to answer the question, but I think it's very important.
Mhm.
Um,
Okay.
Instead of like what are the learning styles? My answer is, I think that most people are misled, or like one of the biggest myths is that there are in fact specific learning styles. So one of the most popular ideas is this kind of VAK, that there's visual learners, there's
Mhm.
auditory learners, and that there are kinesthetic learners, right? So kinesthetic being like the act of doing. And
Mhm.
that, for example, someone who's visually a visual learner, they're going to learn best by, for example, watching, I don't know, watching videos or like looking at equations. Someone who's auditory, right? Maybe they're going to do best like listening to podcasts. Um, That is more or less completely incorrect.
Mm-hmm.
And I think is very misleading to both educators and to students. And I'll give a reason for the reason for why this is. So the first is, as humans, we tend to be really bad at utilizing our own intuition to describe things about ourselves. And so in this case, if I were to ask you, right, do you think you are a visual learner, a kinesthetic learner, or an auditory learner, right? And let's say I asked 100 people. Now, by chance, you have like a 33% chance of being right. Now, right meaning that you would score at some scale on a test.
Mm-hmm.
The actual percentage is around 44.6%. So people are operating your ability to say what kind of learner you are is barely above chance, is in fact not statistically significantly different from chance. Um, Additionally, if I then give you a test that says, okay, you're a visual learner, auditory learner, a kinesthetic learner, and then I actually ask you to do visual, auditory, kinesthetic tasks. So visual could be you watch a movie, and then after I have to give you a recall task where you tell me what has happened in the movie. Or for auditory, it's like, let's say you're listening to a story and you have to then recall what that story is. And then kinesthetic being I blindfold you and I give you a certain set of geometric shapes and you have to feel them. And then I ask you to draw what those shapes look like. And I correlate, your test on this VAC test, sorry, your score on this VAC test to how well you do. They're completely unrelated. So if you
Mm-hmm.
score really high as like a kinesthetic learner, for example, that's actually not predictive at all about how well you're going to do at this actual kinesthetic test. To not to not answer your question, I think there are some misleading ways of like learning styles. Now,
Right.
generally, my advice, not as a neuroscientist, now I'll actually directly answer your question. Um, my advice, not as a neuroscientist, but as a student who, has been studying for many, many years, I think part of learning style really comes down to, of course, objective styles about how people chunk information and how it's good to memorize things. But then a lot of it is just self-directed, right? If you enjoy listening to podcasts and right, you feel motivated to do these things, then you should do that, right? If you enjoy like watching interesting YouTube videos, then you should do that, right? Um, and I think a lot of it should just be intuitive. Do what works for you and what you enjoy, because ultimately that's what you're going to have motivation to do.
I think that's a great way to answer the question too, because yeah, I'm yeah, learning styles being a myth is really something quite surprising because even in like a standard education system that is being fed into students, telling
Hmm.
people that some people are visual learners, they should watch a video, for example,
Yeah.
but yeah, that what you're saying makes a lot of sense, especially it
Yep.
helps make it less abstract and make it more practical for people. So I just want to go over one last thing before we wrap up about
Hmm.
memory. So we all know that younger children have the incredible ability to learn languages, skills, and techniques
Hmm.
quickly. So this ability, of course, diminishes over time and with age. So what is making them so good at learning so fast and efficiently at that young age? What's different about the brains of somebody who's, for example, a child versus an adult versus somebody who's a lot older?
Hmm. Yeah, this is a really good question. I think I've gotten asked this a lot. So what you're describing specifically with language is a phenomenon which in neuroscience we call a critical period, also developmental psychology and psychology.
Hmm.
It's just called a
Yeah.
critical period. Um, it's a period of development in which your ability to do certain things is enhanced. And it's not only language learning, right? There's a critical period for vision, there's a critical period for uh, well, I think it's also multifaceted. So language, it's not only your ability to memorize things, it's also your ability to vocalize and enunciate in a certain way. So the reason that, for example, in high school, I had a Spanish teacher who her Spanish was her second language, but she was perfectly fluent in Spanish and she had been speaking it for decades. That being said, she still will never be able to sound like someone who is native. So whether that's how some people can, for example, roll their R's, and that is due to a critical period in literally how the vocal muscles are tuned to specific words. So developmentally, right, there are critical periods in which your ability to learn things and do things is enhanced. That's why people can become musical savants and learn skills. Well, why is this? Why does this happen? Um, the reason this happens is because the brain is particularly plastic at this age. We just talked about neuroplasticity, and that means that the brain is particularly it's in a particularly good state to change what it's doing. A good example of this is if you're a child, I could literally, so there are cases where it's surgically required to, for example, resect large parts of the brain. Or there are people who are born with only one hemisphere, right? If I reach into your brain right now, I don't know how old you are, but if I reach into your brain right now and pull out your right hemisphere, you would die, right?
Mm-hmm.
Most people would die.
Yeah.
If you do that in a developing infant, they would actually still survive. And the reason is yeah, the brain is in a state where it's particularly plastic. The number of synapses that the brain has is far greater, and there's still development of all of these processes. They're still ongoing. So all of for example, the vocal muscles for specific speech patterns are being developed and refined over time. And that's really why like children have fantastic ability to learn new things in a way that adults don't.
Right. I think it's a great way to explain it, especially I didn't realize that there was also a more physiological reason because of how the children are in development and even their muscles are also
Hmm.
adapting and contributing to all of this. So I
Hmm.
think, yeah, that really answered a very big question that people had and an observation that went unquestioned for many. Lucas, thank you so much for being here.
Yeah.
I think you broke down learning and biological levels and made it feel less like magic and more like something that we can genuinely shape and that is practical. So, for everybody listening, if this episode gave you a new perspective on how the brain learns, share it with somebody who studies, teaches, or is just trying to build a new skill. Because learning isn't random, as we talked about. It's neurons adapting to experience. And every single time you try again, you're not starting from zero. You're starting from a brain that's already changed. If you guys enjoyed this episode and learned something new, make sure to follow on your favorite podcasting platform or on YouTube. I'm Avnish, this is The Early Perspective, and I'll see you in the next episode.